I'm the clinical research fellow at the Center for Genetic Muscle Disorders. I work with Dr. Catherine Wagner, who is the director of our center, and she does send her regards and apologizes that she could not be here in person today. But June had asked me to give a talk about updates in research in FSH, which is an extremely exciting field for me. And in the 3 years that I've been a clinical fellow here, there really has been extraordinary progress made in the field of FSH research. I'm not going to be able to talk about all of it here in the next 45 minutes, but I do want to focus on some of our research efforts, which we're very familiar with, and talk about a few opportunities for patients and families to participate in research. So our research group, the Center for Genetic Muscle Disorders at Kennedy Krieger, is part of a consortium of centers called the Wellstone Muscular Dystrophy Cooperative Research Center. It's sponsored by the National Institutes of Health, and there are 6 of them, actually, and each is a multi-group collaboration where multiple centers get together and investigate projects, each having to do with a specific type of muscular dystrophy. There's one for myotonic dystrophy, there's one for Duchenne. And ours specifically deals with biomarkers in FSH. So biomarkers are measurable indicators of disease. They are usually quantitative. They tell you how likely you are to have disease versus not disease. An example would be like cholesterol. Cholesterol is something that you measure, but what we're really interested in when we're measuring cholesterol is what is your risk of developing coronary artery disease or stroke. And we're interested in biomarkers because they give us clues about what happens during the disease and why it causes muscle pathology. And a secondary reason is that we're interested in biomarkers is that they can be used as outcome measures in clinical trials. This is particularly important in the FSH field because unlike some other forms of muscular dystrophy and other diseases, there really is no good mouse model yet. There was actually a couple of weeks ago, actually, there had been a report that came out in PubMed about the development of a potential mouse model. But up until then, there really has been no other way to test new drugs and new compounds in a mouse model, which is usually required for the development of therapy. The major project of our FSH Wellstone is our biopsy program, which was developed to collect muscle tissue in a systematic fashion in large enough quantities so that multiple biomarker studies can be done in parallel. And we consider this a major success of our program. I'll tell you a little bit about the study itself. The subjects in the biopsy study are enrolled in groups or families. They're called cohorts. And each cohort contains one person who has FSH and one person who does not have FSH. And that person is a first-degree relative. It is a parent, a sibling, or a child. And each subject gets 2 muscle biopsies, and that allows us to collect samples of a muscle that's usually weak in FSH and a muscle that's usually spare or not weak in FSH. And so for this reason, that's what I write. So we collect 4 to 4 plus strength in the weaker muscles, and so that's actually pretty strong. On a scale of 1 to 5, 4 is actually is weak, but not severely weak. And the reason we want that specific range of strength is that we want muscle that is affected, but not completely dystrophied or replaced by scar tissue. And doing the study this way, it not only collects a lot of data, but it allows us to make a lot of different types of comparisons. Within one person, we can compare affected versus non-affected muscle. But we can also, within a family, compare affected versus unaffected individuals. And that has proven to be extremely important because of this idea of family effect. There are genes that are abnormal in FSH compared to people without FSH. But there are also characteristics in genetics that are specific to families, where there are abnormalities, not necessarily abnormalities, but alterations, distinctive features in families that that you don't find in other groups that have nothing to do with FSH. And that could go, that could be divided by ethnicity. That could, and it makes sense because everybody is different genetically, and we're more similar to our family members than not. And having the first-degree family members involved in the study will allow us to subtract a lot of the noise that is due to factors, genetic factors that are not related to the FSH. So a little bit more on the logistics of this. This is the way that the study visit goes. Some of you have participated in this study, and so hopefully we'll have a chance for you to share what that experience was like. This is usually a 2-day process. Usually our study coordinator is the first person to contact a patient, and that is Jenny Bubot, who's in the back of the room. She will do the preliminary screening, see who meets the inclusion and exclusion criteria for the study. And then each subject comes to Baltimore for a 2-day study visit. And on the first day, we do the consent process, and we do examination, and we collect strength data. And we also do a number of labs, including confirmatory testing for FSH, as well as tests to make sure that there aren't any bleeding risks or infection risks related to that could be relevant when getting the biopsy. The second day is biopsy day, and so each person is admitted to our outpatient OR at the Johns Hopkins Outpatient Center. And then we perform 2 muscle biopsies, one on the deltoid muscle, which is high up in the top of the shoulder, and the other is the biceps muscle, which is further down in the arm. The entire procedure takes about an hour per person, and we usually, if there are 2 people, we'll just do them one at a time, one right after the other. If there are 3 people, one right after the other. And afterward, the muscle tissue is collected, and we give it to our technician, and it is shipped off to a number of different centers that all are performing different projects in FSH. So, okay. So the muscles that we collect are actually the ones that are highlighted in red. And unfortunately, I couldn't get photos that had the red both on one side, but we do just one arm. We don't do biopsies of both arms. And the muscle that we collected is distributed in many different directions. You know, it goes onto, it's frozen, it is fixed, it is sent off on a FedEx truck somewhere, even before most of the people are out of the OR. So there have been major efforts in looking at this one here, gene expression, looking at different genes that are expressed in FSH or suppressed in FSH. Looking at proteomics deals with looking at the population of proteins that are in the muscle. I guess I should explain, xenografting is looking at the building of transplanting muscle into animal models, in our case, mice. One of our biggest efforts, however, that I did want to highlight is the cell culture and immortalization project, where we transform the cells into colonies that will continue to divide generation after generation. This forms a large tissue bank where colonies of cells are available for distribution to scientists who basically call us and ask us for FSH tissue. So in this case, I think that that's really exciting because these colonies can be used in experiments that haven't actually been thought of yet, you know, and they can go on to help and to supply future research projects that as we learn more. So currently, our enrollment looks like this. There have been 36 families that have been enrolled, and that's 85 individuals, and each of those individuals has gone either 1 or 2 muscle biopsies. And so this is a large sample. For any group of people with FSH, that's quite a large sample, and we're continuing to enroll 1 to 2 patient groups per month, which is actually a pretty good rate for our capabilities and the other Wellstone. And initially we had planned to only to enroll 30 families, but we really had a very enthusiastic response, and we've been so productive that we've actually expanded that. And so I see some people who have actually participated in this trial, and I wanted to really thank them for their contribution. And if you know of other people who may be interested in participating in this trial, I'll include contact information at the end of this talk. Here's a sampling of some of the publications that have resulted from research in the FSH Wellstone. The first one actually, it might be hard to read, but that actually just came out a couple of weeks ago, and it actually is kind of a pretty interesting study. I don't want to get too far into the molecular biology of it, but it has to do with telomere length. And telomeres are genetic code that are at the very, very ends of the chromosome. And what's very interesting about that is the deletion that causes FSH is actually at the very, very end of chromosome 4. And what this study is showing is that the length of the telomeres, which changes based on how many times the muscle cell divides, is related to the expression of a protein called DUX4, which, as many of you I'm sure are aware of, has been implicated as a major factor in the development of FSH.